Good morning, students. I am Dr. Swati Gangopadhyay, Associate Professor, Department of Microbiology. Today I will cover the topic transcription in prokaryotes. And this is under UG CBCS Semester 3 in Microbiology under the curriculum of West Bengal State University. The paper code is MCBA Core 070 Unit 3. Transcription is the process of synthesis of RNA chain from a segment of DNA or gene. The transcription process is initiated by the enzyme RNA polymerase that actually initiates the formation of phosphodiester bond between the two incoming ribonucleotide triphosphate. The RNA polymerase moves along the template, the RNA chain elongates, and ultimately the RNA polymerase reaches at the end of the gene we call the terminator site, and the RNA chain or the newly synthesized RNA chain we call it the nascent RNA that is separated from the DNA template. So, it is the complementary strand or the sequence that is complementary to the newly synthesized DNA is acting as the template strand, and the other strand whose sequence is similar to the nascent RNA is the coding strand. The coding strand only differs in sequence from the nascent RNA in that the uracil is incorporated in place of thymine. So, in this diagram you can see the double stranded DNA. The template strand is represented by the black color and it is shown from 3' to 5' direction, and the coding strand in red color that is in 5' to 3' direction. We always know that either during the replication process or during the transcription process, the synthesis of DNA or synthesis of RNA follows the polarity from 5' to 3' directions. Now, you can see the RNA sequence is complementary to the template strand and that is equivalent to the coding strand, except uracil is incorporated in place of thymine as like the first base from the 5' end. So, in this way the new RNA transcript is being made. Now, we can define the transcription unit as follows. So, this is more or less a rough schematic representation of a gene. The gene has basically the start point region and a regulatory region that is lying at the upstream portion, and the functional portion of the gene lying at the downstream portion, that is in the right hand side of the start point, and the promoter sequence is situated at the left hand side of the start point. So, the region left hand side of the start point we call it the upstream region or the upstream sequences, and the right hand side from the start point we call it the downstream sequences, that is indicated by the positive signs, and the upstream sequences are indicated by the negative signs. The sequences those are lying basically towards the upstream region is known as the proximal region, and the downstream regions are indicated as the distal region. Now, you can see in this diagram that RNA polymerase basically recognizes the promoter sequences, initiates transcription from the start point, ultimately it reaches to the terminator or the end of the gene, then the RNA polymer is dissociated out and the transcript is released from the DNA template. So, RNA synthesis is basically catalyzed by the enzyme RNA polymerase. Transcription starts when RNA polymerase binds to a special region, that is the promoter sequences at the start of the gene. The promoter basically surrounds the first base pair that is transcribed into the RNA, and we call it the start point. From this point, RNA polymerase moves along the template, synthesizing the RNA until it reaches to the terminator sequence. This action basically defines transcription unit that extends from the promoter to the terminator sequence. And in case of bacterial system, we know their genome is polygenic, so many proteins are coded from the gene, those are lying side by side. So, for that cases, the transcription unit may include more than one gene. I have already described in the earlier diagram the upstream and the downstream sequences, and by this we can easily understand that the RNA that is being transcribed have a usual direction from 5 prime to 3 prime end. Now, basically RNA synthesis occurs in the transcription bubble. So, when the RNA polymerase binds to the double stranded DNA, the double stranded DNA will undergo melting. We call it the strand separations, and both the strands will be separated out. Here you can see in this diagram, one strand, that is the coding strand, is represented by the green color, and the template strand is represented by the blue color. So, these strands will be separated out, that is called the unwinding, and the first base appear at the start point region, and the phosphodiester bond is being formed with the successive bases, and there is the initiation of RNA synthesis. So, the immediate product of transcription is called the primary transcript. It would consist of an RNA extending from the promoter to the terminator, possessing the original 5 prime and the 3 prime ends. However, the primary transcript is almost always very unstable for the bacterial system mainly, and we have seen in eukaryotic system this primary transcript is basically undergo some sort of modifications, that means the 5 prime capping, 3 prime polyadenylation, and also the splicing events occur, so that the mature products are being produced. But in case of bacterial system, mainly the messenger RNA, that means the RNA from which the proteins are being produced, they are very much unstable, and for that in bacterial system or in prokaryotic system, the transcription and translations are coupled process. So, following transcription, translations are being initiated from the 5 prime end of the messenger RNA until the 3 prime end of the transcripts are being produced, or before the 5 prime 3 prime end of the transcripts are being produced. And this in prokaryotic system, this messenger RNA is very rapidly degraded, or the other RNA other than the messenger RNA, there are the tRNA and the rRNA. tRNA is absolutely required for the translation process, and rRNA that makes the ribosome. These RNAs are produced, and these primary transcripts are cleaved or spliced in some certain regions to give the mature products. The transcription is the first stage of gene expression, and it is basically the principal step at which the gene expression is being controlled. Now, if we consider the rate of transcription, then the overall rate of transcription is basically 40 nucleotides per second at 37 degrees centigrade. So, you can see in this diagram that RNA polymerase binds to the promoter region, initiates the formation of the transcripts or initiates the formation of the phosphodiester bond. The length of the RNA chain is being increased by the subsequent additions of the nucleotides. The transcription complex moves along the DNA template until it reaches to the terminator sequences. So, definitely this catalytic reaction has a speed and the rate of transcription is 40 nucleotides per second at 37 degrees centigrade, and this is almost same as the rate of translation, that is 15 amino acid per seconds, but the rate of transcription is much more slower than DNA replication that occurs at a rate of 800 base pair per seconds. Now, the RNA polymerase moves along the DNA, the transcription bubble moves with it and the RNA chain grows longer. The process of base pairing and base addition within the bubble is catalyzed and also it is scrutinized by the enzyme itself, that is by the RNA polymerase. The 3' hydroxyl group of the last nucleotide added to the chain reacts in an incoming nucleoside 5' triphosphate. That means the phosphodiester bond is formed between the 3' hydroxyl group of the last nucleotide base in the RNA chain and the 5' phosphate, that is the alpha phosphate. We know that nucleoside triphosphate basically has 3 phosphate, alpha, beta and the gamma phosphate. The alpha phosphate is involved in the phosphodiester bond formation reactions and the beta and the gamma phosphate is being liberated out as pyrophosphate, and this pyrophosphate later is cleaved with the enzyme pyrophosphatase to generate 2 inorganic phosphate. The RNA polymerase moves along the DNA and the transcription bubble moves with it, as I have already said. Now, we can divide the entire transcription process in 3 stages: initiation, elongation and termination. So, first the process is the initiation, and during the initiation process the most crucial part is the template recognition at a specific sequences, that is at the promoter sequences. So, DNA starts when the RNA polymerase binds at the promoter sequences, the DNA starts unwinding at the promoter region and the initiation of the phosphodiester bond formation starts. So, what happens at that time? The RNA polymerase basically does not move during this period. It remains associated tightly at the promoter sequences and it will try to form the phosphodiester bond between 1, 2, 3, 4 nucleotides, but it will not be successful to add more nucleotides, and the small RNA nucleotide chains, maybe it is of 2 to 3 or 6 base pair in length, they are coming out from the template. We call it abortive initiation. Why this abortive initiation takes place? Because there are some geometrical configuration that is occurring due to the binding of the RNA polymerase at the promoter sequences, and that configuration is not adequate enough at this particular period to hold the RNA chain, and the RNA chain, short RNA chain or the short RNA fragment, rather it is better to say, they are coming out as the abortive transcript. But the RNA polymerase will try to make the transcript longer. So, there will be some changes in its conformation and ultimately the RNA polymerase wins and the transcript gets increased in its length and the complex will be converted into the elongation complex. That means that it will be a much more stable complex, and the RNA polymerase, DNA and the nascent RNA chain starts leaving the promoter. We call it the clearance of the promoter, and the RNA polymerase will move through the DNA template and it reaches at the termination sequences, and the RNA chain will be released. Now, if we see the bacterial RNA polymerase, then we will see in E. coli system the RNA polymerase is a quite big enzyme, and its molecular weight is almost 465 kilodalton, but it is not a single subunit protein. It is a multimeric protein, and this is made up of basically 4 to 5 subunits. So, you can see here there are 2 alpha subunits. This alpha subunit, its alpha subunit has the molecular weight 40 kilodalton each, and this alpha subunit is produced from the gene, that is the RPOA. Then comes the beta subunit. Beta subunit has the molecular weight 155 kilodalton, and it is produced from the gene, that is the RPOB. Next is the beta prime subunit. It is the biggest subunit. The molecular weight is 160 kilodalton, and it is produced from the gene, that is RPOC. So, this beta and beta prime subunit, they actually forms the catalytic center or the catalytic core of the RNA polymerase. So, what is the functions of the alpha subunit? The alpha subunit basically helps the enzyme assembly at the promoter sequences, and also they binds to some activator, and they also have some role in recognizing some portion of the gene that is located further upstream of the promoter sequences. So, this catalytic center, and when this catalytic center, that means the beta and the beta subunit is associated with the alpha subunit, we call it the core part of the RNA polymerase. So, this is the core RNA polymerase. Other than the core polymerase, there is another subunit, that is the sigma subunit. Molecular weight is 32 to 90 kilodalton, and this sigma subunit has enormous significance in transcription. When this core complex joins the sigma subunit, we call it the holoenzyme complex. So, what is the functions of the sigma subunit? Sigma subunit basically gives promoter specificity or the promoter recognition, at which promoter specifically the RNA polymerase holoenzyme complex will bind. Now, suppose a bacteria that is very much infective to any individual, like Mycobacterium tuberculosis, that causes the tuberculosis to us. So, now people tried, or scientists tried, can we have design some mechanism, or can we have get some antibiotic that can target the any subunit of RNA polymerase, specifically that bacterial RNA polymerase. So, the bacterial transcription will be stalled, but the host transcription will not be get altered. In this way, we can kill the Mycobacterium tuberculosis. So, there is one drug, that is the rifampicin. It is widely used in the treatment of tuberculosis or Mycobacterium tuberculosis, and this rifampicin actually binds to the beta subunit of RNA polymerase and prevents the beta subunit in the progression of the transcription. So, prevents the beta subunit from its action, and it blocks the initiation of the transcription mainly prior to the formation of the first 2 phosphodiester bond. If the phosphodiester bonds are still being made, then rifampicin inhibit the actions of beta subunit in such a way that it prevents the RNA chain from extending beyond 2 to 3 nucleotides, and thus blocks the bacterial transcription or inhibit the transcription in Mycobacterium tuberculosis, as I have cited the example. Now, there is an interesting process how the RNA polymerase binds to the DNA. So, I have already told you that we can consider RNA polymerase in two ways. One is the core enzyme and another is the hollow enzyme. Now, the core enzyme has a great affinity for the DNA. It can bind anywhere on the DNA. So, basically the DNA is negatively charged, you know, there is the phosphodiester backbone and there are some positive amino acid residues on the surface of the RNA polymerase. So, there will be a good affinity or there will be a very good binding, very good friendship between the DNA and the core enzyme complex, and they can bind at anywhere on the DNA. We call it the loose binding. But one thing you should remember that it is not at all desirable to bind the RNA polymerase at anywhere on the DNA and starts transcription from any non-specific sites. We know each and every gene has a certain regulatory sequences, that is the each and every gene has a certain promoter sequences, and that promoter sequence is very much specific for that particular gene only. So, RNA polymerase has to be recognized that particular promoter and initiates transcription from the specific start point of that gene. So, it is a very specific type of initiation. So, who determines that? That is the sigma factor, and it is acting just as a searchlight or a torchlight. It will focus where the RNA polymerase has to be installed. Then the sigma factor comes, joins with the core complex, and the hollow enzyme structure will form, and all the RNA polymerase that was bound loosely at any sequence of the DNA will be dissociated out, and only it will bind to the specifically to the specific promoter sequences. So, sigma factor basically governs the specific type of localization of RNA polymerase at the particular promoter sequences. So, the sigma factor is usually released when the RNA chain reaches 8 to 9 bases. What does it mean? It means that the hollow enzyme initiate transcription from the specific start point or from a specific promoter sequences, because it binds very stably and very tightly to the promoter regions. Now, it will start initiation. After abortive initiation, there will be the elongation complex. The sigma factor's role is over at the initiation site. When the RNA chain will be elongated at a length of the 8 to 9 base pair, then the sigma factor will be dissociated out from the hollow enzyme complex, and the core enzyme is sufficient enough to synthesize the RNA on the DNA template. But the core enzyme cannot initiate the transcription at the specific locations, that means from a specific promoter sequences, and for this the assistance of sigma factor is very much needed, so that hollow enzyme binds to the promoter sequences and initiates transcription from the start site, which is surrounded by the promoter sequence. Now, the core enzyme has a general affinity for the DNA, that I have already told you. There is the electrostatic interactions between the basic protein and the acidic nucleic acid. So, any random sequences of the DNA will be bound by the core polymerase. We call it the general binding reaction, and we also sometimes refer it as the loose binding. The sites are regarded as the loose binding sites. No change occur in the DNA and it remains in the duplex condition. So, there will be no unwinding or no melting reactions at that specific loose binding sites. Moderately the complex is stable at that sites. They have the half life for dissociation nearly 60 minutes. The core enzyme does not distinguish between the promoter and the other DNA sequence. For the core enzyme, it is same for all the sequence of the DNA. Now, when the sigma factor comes and joins into the core enzyme to form the hollow enzyme complex, it introduces a major change in the affinity of the RNA polymerase for the DNA. So, the holoenzyme has a drastically reduced ability to recognize the loose binding sites, that means at any general binding sites, and their association constant is reduced at that site to a factor of 10 to the power 4, and the half life is very much reduced. So, it will come down to nearly less than 1 second. So, sigma factor, what it does? It basically destabilizes the RNA coenzyme complexes at any random sites, but simultaneously sigma factor does another functions. It confers the ability to recognize the specific binding sites with a very high association constant, and the association constant will be increased for that enzyme almost by 1000 times, and half life will be for several hours. So, it is a very tight binding, very specific binding and very stable binding. So, this is represented in this diagram when the holoenzyme binds to the DNA, it will bind with the equilibrium constant 10 to the power 6 to 10 to the power 9 minus 10 to the power minus 6 to 10 to the power minus 9 molar. Then the DNA melting will start, and the rate constant for this reaction will be 10 to the power minus 3, 10 to the power minus 3, 10 to the power minus 1, second minus 1. Abortive initiation takes place, it will association constant will be K1, 10 to the power minus 3, second minus 1. Then there will be the promoter clearance and the release of the sigma factor that occurs at a rate of greater than 1 to 2 seconds, and RNA synthesis begins. RNA polymerase binds to the promoter sequences, then first the closed binary complex is being formed. Binary means there are two components, one is the RNA polymerase holoenzyme and another is the double stranded DNA. Next step is the melting of the DNA and open binary complex is being formed. So, open because there is the melting reaction that has been started, and the RNA polymerase is associated with this open region of the DNA. So, it is the open binary complex. Now, when the first phosphodiester bond formation takes place, abortive initiation takes place, then the RNA chain has been just initiated. So, three components are there, the opened DNA, the RNA polymerase and the newly synthesized RNA chain. We call it the ternary complex, and ultimately when promoter clearance takes place, the RNA polymerase leaves the promoter, further moves to the downstream sequences, then we call it the elongation complex. Now, coming to the abortive initiation, after each base is added, there is a certain probability that enzyme will release the chain. This comprises an abortive initiation, after which the enzyme begins again with the first base. A cycle of abortive initiation usually occurs to generate a series of very short oligonucleotide that has been very well demonstrated experimentally. When the initiation succeeds, there is no requirement for the sigma factor, or it is not at all necessary, and I have already told that the enzyme makes the transition to the elongation ternary complex of the core polymerase DNA and the nascent RNA chain. Now, the critical parameter here, how long it takes the polymerase to leave the promoter so that another polymerase can initiate. So, it is very crucial. If the RNA polymerase core complex leaves the promoter first, then the chances of reinitiating the transcription process in the another promoter sequences by this same core polymerase will be higher. So, it will enhance the rate of transcription. The parameter of the promoter clearance time, its minimum value is 1 to 2 seconds. The enzyme then moves along the template and RNA chain extends beyond the 10 bases. So, it is now a very much stable complex, and the RNA polymerase will continuing transcribing the RNA chain. Now, in this diagram we can see the locations of the sequences that is being covered by the initiation complex, initial elongation complex and general elongation complex. So, 1 is the start point, plus 10, plus 20, plus 30 are the downstream sequences, and minus 10, minus 20, minus 30, minus 40, minus 50 are the upstream sequences. So, when the holoenzyme binds the RNA polymerase, it will cover a bigger region of the DNA extending both upstream and the downstream sequences from plus 20 to minus 50. And this contains the sigma factor and almost covers the region up to 75 to 80 base pair. Now, when the initial elongation complex forms, that means the just RNA chain has been initiated, then the RNA polymerase holoenzyme will lose the sigma factor and the contact site of the RNA polymerase or the contact region of the RNA polymerase is reduced. It will extend from plus 20 to minus 40. So, almost minus 35 to minus 55 base contacts will be loose. Now, when the complex is converted to the general elongation complex, it forms at 15 to 20 bases and covers 30 to 40 base pair, mainly minus 20 to plus 20 region. Now, coming to the promoter structure, there are 4 conserved features in the bacterial promoter, that is the start point, then at the further upstream minus 10 sequences, minus 35 sequences, the separation between the minus 10 and the minus 35 sequences. So, all 4 regions are very much important. Now, what is the nucleotide present at the start point? For more than 90% cases it is the purine. It is common for the start point the central base be the CAT. So, one adenine that is flanked by 2 pyrimidine residue. Adenine is the purine, cytosine and thymine is the pyrimidine residue. Just upstream of the start point, a 6 base pair region is recognizable that is present in almost all promoters for the genes, and this we call the minus 10 sequence. Earlier it is called the Pribnow box. It is very close to the 10 base pair upstream of the sequence, and also it is known as the Tata box. So, this Tata box has the sequence TA, TA, AT. So, it is AT rich, and we have seen that the melting region starts from the promoter sequences. So, it is AT rich. We know that adenine and thymine forms 2 hydrogen bonds. So, less energy will be required to separate the 2 strand of the DNA by breaking the hydrogen bond in between the thymine and adenine sequences. So, the sequences will be read as TATAAT. The 80, 95, 45, 60, 50, 96 means at what percentage this particular nucleotide at that particular location is being conserved. So, first T, that is the maximally conserved 80%, if we see 100 promoters, for 80% cases we will see the first nucleotide at the promoter is thymine. Then comes adenine, that is the maximally conserved, that is almost highly conserved 95, 30 is less conserved 45, then for adenine 60, next adenine 50 and last thymine maximally conserved 96. So, this is how the gene looks like. This is the start point at the first figure, part 1, that is designated at figure 6.10. I have taken it from the your textbook Molecular Biology Weaver. So, transcription starts from the start point and there is the minus 10 sequences, you can see TA, TA, T. So, small letter actually indicates the that particular nucleotide is conserved less than 50%. And there is another sequence, we call it the -35 box, which is basically separated by 15 to 17 nucleotides from the -10 box. The sequence is TTGA-CA. So, this is the structure of the core promoter. Other than this -10 box and -35 box, for some specific promoters we find some other specific elements. So, you can see in the next part of the figure, the core promoter is designated by the blue box -10 and -35 sequences. Then from the region -40 to -60, there is another conserved sequences, we call it the up element. This is not as conserved as the promoter sequences, but in some promoter like those genes, the genes those are expressing at a high level. For example, the ribosomal RNA, these elements are present, and we call it the up element that extend from -60 to -40 to -60, which is followed by some other element. So, that is represented by the yellow box extending from -60 to -150, and it is shown by 3 boxes 1 and 2 and 3. These regions are acting in some manner which is more or less analogous to the enhancer sequence of the eukaryotic promoter, and some factors binds to this, or some factors or transcription factors binds to these sites. We call it the fish sites, and when these factors binds, they actually stimulate the activity of the RNA polymerase at the promoter. So, I have already explained that. So, what is the definition of the optimal promoter? The optimal promoter is a sequence consisting of the -35 hexamer separated by the 17 base pair from -10 hexamer lying 7 base pair upstream of the start point. So, this is the definition of the optimal promoter, and the separating distance is very important. Separating distance between the -10 and -35 box is very important for recognitions of the RNA polymerase and for binding of the RNA polymerase tightly to initiate the transcription, but exact sequence, intervening sequence is not very much needed. Now, how can we actually measure the efficiency of a promoter? So, some genes are highly expressed in some particular tissue or system in bacterial system, some genes may not be that much efficiently expressed. So, the transcription or the efficiency of transcription actually depends on the efficiency of the promoter. If the promoter are very strong promoter, then the rate of transcription will be high. If the promoter is weak promoter, then the rate of transcription will be low. So, this efficiency of the promoter can be measured by two ways, the promoter down mutations and the promoter up mutations. So, promoter down mutations most are identified in bacterial mutants, in mutant strains of bacteria that have lost or have very much reduced transcription of that adjacent genes. So, if you can introduce some mutation in the promoter sequences of a particular gene, and if that mutation actually causes the rate of causes the reduction at the rate of transcription, then you can designate the promoter as the down mutation, promoter mutation as the down mutation. And if you do just the reverse, that means you are introducing some mutation in the promoter sequences that actually increases the efficiency of the promoter, then you can consider that mutations as the promoter up mutations. Now, I have already mentioned the abortive initiation of the transcription. So, we can skip that. When the initiation succeeds, the sigma is no longer necessary and the enzyme makes the transition complex or the initiation complex to the elongation ternary complex of the core polymerase and the nascent DNA. The critical parameter here is how long it takes for the polymerase to leave the promoter. We have already discussed that. Now, the sigma factor can be recycled. So, in this figure you can see the double stranded DNA, that is the promoter sequence is bound with the holoenzyme complexes. The sigma factor is designated in blue color and the core enzyme is designated in the red color. So, forming of the open complexes, there is the strand separation or unwinding of the DNA, then incorporation of the first nucleotides or the initiation of the RNA chain. So, sigma factor is still associated there, and when the promoter is cleared from the holoenzyme complex, that means the holoenzyme leaves the promoter and ultimately moves to the further downstream of the gene, then the sigma factor will be released out, and this sigma factor can be recycled. So, this means that after initiation of the transcript or after the promoter clearance, when the holoenzyme complex will move further downstream, then sigma factor can be released from the holoenzyme complex. The coenzyme will be sufficient enough to carry out the transcription till it reaches to the terminator, and the released sigma factor now gets assembled with the another coenzyme. It will make the holoenzyme and it may bind to again to the promoter sequences to start the second round of transcription. Now, coming to the structure of the sigma factor. If we see the structure of the sigma factor, then you can see it can be divided into 4 segments. So, segment 1, which is very close to the amino terminal end, and segment 4, which is very close to the carboxyl terminal end. Now, we can divide the segment 1 into 2 parts, 1.1 and 1.2. Segment 2 is divided into 4 parts, 2.1, 2.2, 2.4. Segment 3, 3.1 একটাই আছে 3, one segment is there 3, and segment 4, that is 4.1 and 4.2. এই জায়গাটা একটু কাট করো না, বলবো। Now, we come to the sigma factor. If we see analyze the structure of the sigma factor, you can see this sigma polypeptide, you can see the amino terminal end, which is formed by a region, that is the region 1. It is divided into 2 sub-region, 1.1 and 1.2. Then the second region sigma 2 is divided into 4 parts, 2.1, 2.2, 2.3 and 2.4, sigma 3 and sigma 4, 4.1 and 4.2, which is followed by the carboxyl terminal end. So, what is the functions of the region 1? Region 1 is only found in the primary sigma factor. Primary sigma factor, that is the sigma 17 E. coli or sigma 43 in Bacillus subtilis. So, this primary sigma factors means the sigma factors which is responsible for transcription of mainly the all genes necessary for the physiological systems. These are the primary sigma factor, and there are the other sigma factors which are required for expression of certain genes under stress condition, like heat shock responses, that proteins or the genes that will be expressed when a bacteria is under heat shock, under starvation, during sporulation or nitrogen fixation, like that. So, for that specific type of sigma factor, region 1 is not present. Region 1 is found only for the primary sigma factor, and its role appears to be prevent sigma factor from binding to the DNA. So, if the sigma factor itself along covers the DNA, then there will be no entry of the core polymerase there. So, sigma factor, sigma region 1 actually makes an inhibitory domain that prevent the sigma factor to bind itself to the promoter, and thus it favors the formation of the holoenzyme complexes. Now, the region 2 is found in all sigma factors, and it is most highly conserved region of the sigma factor. Region 2 can be subdivided into 4 parts, 2.1 to 2.4, and the region 2.4, that is actually responsible for binding to the -10 sequences or the Tata box. Now, very little is known about the region 3. One helix turn helix DNA binding motif is there, like region 4, like region 2. Region 4 is also subdivided into 4 parts. subdivided into some subregions, and 4.2, that region is very important. 4.2 region actually recognizes the -35 box of the promoter. Now, there are some channels which are present in the RNA polymerase that actually helps the binding of the template strand or the non-template strand, then binding of the ribonucleotides, then it favors the escape of the RNA chain, newly synthesized RNA chain from the RNA polymerase. So, from the left hand side you can see there are the template DNA, there is one strand is the template strand and another is the non-template strand, and in the purple color you can see there is the RNA exit channel, the RNA newly synthesized RNA exit from this channel. There is the beta flap, and you can see the different subregion of the sigma 3.1, 3.2 region. Then in the yellow box it is designated sigma 2 region, that sigma 2.4 region binds to the -10 sequences, and you can also see at the downstream region the sigma 1 region. There is another channel from where the non-template strand enters into the sigma factor, and another T channel, through the T channel the template strand enters into the sigma factor. So, there are basically 5 channels into the enzyme. The NTP uptake channel allows the RNA NTP or the RNTP to enter the active center, that means that means at the core of the beta subunit. The RNA exit channel allows the growing RNA chain to leave the enzyme it is synthesized during the elongation. The remaining 3 channels allow the DNA entry and exit from the enzyme. These are the downstream DNA channel, non-template strand channel and the template strand channel. So, this is almost about the initiation of the RNA transcription or the initiation of the transcription process. Now, we will come to the elongation phase. So, during the elongation phase, the double stranded DNA enters the enzyme through defined channels and the strands are separated. The RNTPs enters the active sites to the defined channel and are added sequentially to the complementary base pair on the template DNA with the hydrogen bond, and phosphodiester bonds are simultaneously formed. Only 8 or 9 of the growing RNA chain remain base paired. So, how many RNA nucleotide will be base paired with the template strand? That is mainly 8 to 9 base pair, rest are escaped through the RNA channel, RNA exit channel. Now, there are we know that DNA polymerase has a very good proofreading activity. If some altered or defective base or altered, I means wrong base are incorporated, then the DNA polymerase can scrutinize during the replication process with its 3' 5' exonuclease activity and removes the erratic base and replace the errored base with the correct bases. But RNA polymerase does not have that much of proofreading activity. That is why we can see that viral RNA that are virus that are using the RNA as their genome, when they are replicating their RNA with the help of the RNA polymerase, then wrong bases are frequently incorporated, and for that reason new strains of viruses are quickly emerged. So, but still there are some sort of proofreading activity in the RNA polymerase. The enzyme uses its active site, that means the catalytic site which is lying at the beta center, beta core, in a simple breakdown to catalyze the removal of an incorrectly inserted ribonucleotide by reincorporation of PPI. So, what happens during the phosphodiester bond formation? The beta and the gamma phosphate will be liberated out in the form of the PPI, that is pyrophosphate, that is later cleaved by the enzyme pyrophosphatase. But during this scrutiny program, the RNA polymerase can reintroduce the PPI again and the correct and next the enzyme can incorporate another ribonucleotide in its place in the growing RNA chain. So, it is called the pyrophosphorylated editing. Now, how the elongation complex moves? There are basically 2 hypotheses. In one hypothesis, the first one you can see that the double stranded DNA is in the blue color and the newly synthesized RNA is in red color. The RNA is completely wrapped over the double stranded DNA, because the RNA polymerase is moving around and around to the double helix as indicated by the yellow arrow. So, these actually avoids the straining of the DNA, but it wraps the newly synthesized RNA product around the DNA template. So, this is one hypothesis, and another hypothesis is that RNA polymerase moves in a straight line. So, it is just moving in the straight line, and when it is moving in the straight line, the newly synthesized RNA chain is associated with the template DNA for 8 to 10 bases, and the rest of the DNA will be liberated out from the template DNA by the RNA polymerase exit channel. But while adopting this mechanism, two phenomenon occurs. So, what happens, the the downstream part of the RNA polymerase or the front part of the RNA polymerase, the strands has to be unwinded so that the RNA polymerase can move. So, strains will be generated on the upstream portion and it will be further supercoiled. So, there will be a change in the superhelical density, and obviously to remove the superhelical density or to maintain the superhelical density in proper way, the topoisomerase has to play a major role, because there will be the twisting of the DNA strands at the back of the RNA polymerase, and the RNA polymerase has to untwist and that strands has to be untwisted. Now come to the termination phase. When the RNA polymerase reaches the terminator sequence at the end of the gene, it will actually fall off from the template and releasing the RNA. E. coli cell contain about equal numbers of two kinds of terminators. The first kind, known as the intrinsic terminator, that functions with the RNA polymerase by itself without the help of the other proteins, and the second kind of terminator, that is known as the row dependent terminator, and these actually require the involvement of an auxiliary factor called rho. Naturally, these are called row dependent terminators. So, coming to the process of row independent termination, row independent termination or intrinsic termination depends on the terminator consisting of two elements. So, this type of termination absolutely depends on some specific sequences or the template strand that is necessary for the termination process, because this type of specific sequences will be automatically, complementary sequences will be automatically come to the RNA chain, and that will form a typical structure of the RNA, not the linear structure, we call it the hairpin structure, and this hairpin structure is absolutely needed for the termination process. The model of termination depends on the hairpin structure in the RNA transcripts at that inverted repeat. So, we can see this picture. So, inverted repeats, we know if we see the sequence from the 5 prime to 3 prime sequences, TACGAAG, TTCGTA, 5 prime to 3 prime. So, what nucleotide is present at the center? It is the G which forms hydrogen bond with the C. So, if you read the sequence of the both strand from the 5 prime end, the sequence will be same around flanking the center base, that is the TA upper strand. If I read from the 5 prime to 3 prime direction, it is TACGAA, and the lower strand if I read from the 5 prime to 3 prime direction, it is TACGAA. So, we call it the palindromic sequences. Now, if the RNA is produced from the 5 prime to 3 prime sequences, then we will get the sequence like this: UAC, GAA, center G, then UU followed by CGUA. So, U will be incorporated in place of T, and this if we carefully notice this sequence, then you will find that the first U and the last A, that is complementary. So, it will make hydrogen bond. Second A from the left hand side and the second U from the right hand side are complementary, it will form hydrogen bond. Third C from the left and third G from the right is complementary, it will form hydrogen bond. In this way, the hydrogen bond will be formed for 1, 2, 3, 4, 5 nucleotides. The A and the U at the apex of the hairpin cannot form the base pair because of the physical constraints of the turn of the RNA. Last AU and the G, the AU cannot form the hydrogen bond because of the physical constraints that is appearing during the formation of the turn of the RNA, and it will form a hairpin structure like this. So, this is mainly the structure of the hairpin. If we see you can see there in the pink box there is a GC rich region. So, GC means there are 3 hydrogen bonds and this is a very stable hydrogen bond. Then you can see the loop structure, hairpin loop UAG, UAG, UAG like this, and the stem, and the end of the hairpin you can see are stretches of U residues. So, this is very important, this stretches of U, presence of these stretches of U residues is very important, and the template strand there is the A residues, they are at the U base pairing, and the length of the hairpin will vary from 7 to 20 base pair. So, the typical distance between the hairpin and the U regions are 7 to 9 base pairs, and there is the GC rich sequences. Probably all hairpins that form in the RNA product cause the polymerase to slow and perhaps to pause the RNA synthesis. Now, in the downstream sequences, we find that there is a U region, and this U region destabilizes the RNA DNA hybrid when polymerase pause at the hairpin, and the template there is the A. So, there will be the RU-DA base pairing. R means ribouridine and DA means deoxyriboadenine, and this is the weak base pairing. It requires least energy of any RNA DNA hybrid to break down the association. So, actually the RNA polymerase is getting a pause at the time this type of specific hairpins are being formed. There is a very weakened interactions between RU and DA. Ultimately, it causes the release of the RNA chain at the terminator sequences. So, this is actually the row independent termination, and next or the final part of the story is the row dependent termination. So, row factor is essential protein in E. coli. It functions solely at the stage of termination. It is basically a 275 kilodalton hexameric protein of identical subunit. That means 6 monomers are associated to form the protein. This row protein has an RNA binding domain, that means it has a domain that can bind to the newly synthesized RNA, and it requires energy for its movement to reach terminator sequences. So, ATP hydrolysis is required. Rho is a member of the family of the hexameric ATP dependent helicases. So, it has the helicase actions also. So, it can favor the separation of the RNA from the DNA strand that functions by passing nucleic acid through the whole of the middle of the hexamer. Rho actually forms a shape like a ring, and the RNA chain and it binds or the RNA chain is inserted at the middle, inserted into the whole portion of the ring, and in this way the Rho protein binds to the RNA. So, row dependent terminators account for about half of the E. coli terminators. Almost E. coli have 50% of row dependent terminators of all of its gene. They were discovered in fast genome where they have been most fully characterized. Now, the row's key function is its helicase activity. I have already mentioned. Energy is given by the ATP hydrolysis process. The initial binding sites for row is an extended 70 nucleotide single stranded region of the RNA upstream from the terminator sequences. So, we can see in the diagram that RNA polymerase transcribe the DNA. When it will almost reaching to the terminator sequences, you can see the Rho protein binds to the RNA, that is 70 base pair sequence upstream from the terminator sites. Rho moves along the RNA, following the RNA polymerase already get a pause at the terminator sequences, and Rho catches up that at that time. So, and ultimately with the help of the ATP hydrolysis, Rho unwinds the DNA RNA hybrid and the RNA chain is liberated out from the DNA template, and full length RNA is being transcribed, that may act as the messenger RNA, that may act as the, after processing they may act as the ribosomal RNA or the tRNA. So, that is all for today's lecture. The books, textbooks and the reference books I have consulted: Molecular Biology Weaver et al., Molecular Biology of Gene Watson et al., Genes 8 Benjamin Levin et al., and all the diagrams are taken in this PowerPoint